It warms it up a little bit if I close that door. I mean a little bit. Yeah. I think it's a little warmer in the back if you're freezing. I know it's weird for professors to tell students to sit in the back of the room, but there's probably heat rises, and that's not as close to the front door where all the cold air is coming in. So if you can't feel your fingers, maybe move to the back row. How's that? You'll have a nice week. Last week had a long weekend and then a little bit of a break, kind of leisurely watching the lecture. So I'm going to call it out on the Google Analytics. Some of you are overly leisurely watching that video, and you still need to do it. So I would strongly encourage you to check out that lecture. Okay, we have less than half of the class has actually watched that lecture, according to the stats. So it doesn't tell me who, so it's not like Big Brother's watching that carefully, but it lets me know who's tracking with the videos. There's 2 videos. Did you see that? There's a lecture 1 and a lecture 1.5. That was a lecture. You didn't see it because I think it was in the beginning and I cut it off. But I got way off tangent, so I had to finish the last couple of slides in my office. Any questions before we get started? Any questions? This is going to be a heavy week. Okay, it's going to be a heavy week. It's going to be a busy weekend. And then Monday evening at 5:15, you have the rest of the week off as far as I'm concerned. Okay? So you get the exam done, take off for Thanksgiving. I don't have anything to worry about for my class. So the flip side is we don't have the exam before Thanksgiving. We have the exam after Thanksgiving. And I actually think that's just cruel and unusual punishment if we can avoid it. Because then you have to focus on something over Thanksgiving other than being with loved ones. So that was the rationale. Some of you may like it, some of you may not, but that's what I was thinking, is get it done before Thanksgiving. Now, I can't control your other classes, so maybe you have other work that you have to do over the holiday, but at least for my class, you don't. Question?
Do we have an online lecture for that Wednesday before Thanksgiving, or is it just?
We do have an online lecture, but we're not going to have class. We're not going to post it here. Okay. So you can watch that, instead of football on Friday, if you'd like, or instead of football on Thursday, if you'd like. Okay. Or if your team loses, then watch the lecture. That'll cheer you up. Okay. All right. So you're going to want to participate this week, both today and Wednesday. It's important information. If you haven't seen the lecture yet and you need to get caught up, this is a really good week to kind of just lean in heavily on nerve physiology. A little challenging concepts. We're going to talk about action potentials today. We introduced that last week in the online lecture, but we're going to reinforce that. We're talking about ion movement. And the ion movement shouldn't be new. I mean, we've been talking about it all semester. We talked about it a little bit with muscle physiology. We talked about it with the beginning of Unit 1. We talked about the ATP sodium-potassium pump, otherwise known as the ATPase pump. So we're going to revisit that. We're going to be moving some ions. There's some memorization that needs to happen. So I'm going to encourage you to maybe get some of these cards or do like a digital type of memorization note card. All right, before we get started, what do you get when you cross a vampire and a teacher? Blood tests. Okay. What's a frog's favorite kind of flower? A frog's favorite kind of flower. A crocus. What type of music do mummies sing in the shower? Rap music. Nicely done. Okay, a bonus. What did the brother Cell say to his sister when she stepped on his toe? My toe, sis. All right, explain it to your neighbor if you don't get it. I really like that one. Yeah. Okay. All right. Who's still struggling? Okay. All right. Don't admit it. That's fine. You don't have to admit it. No shame. No shame. See me afterwards. I'll walk you through it. Review sessions. Okay. These are posted. Some of them are posted. The rest of them will be posted. You can write them down. Tuesday, 4 to 5 p.m., Science Annex 117. Thursday, 5 to 6 p.m., Science Annex 122. Thursday, 7 to 8 p.m., WETA 242. And then Sunday afternoon in this room, Bio 265, 3 to 4. Okay? So you should know by now how this works, but just to reiterate, these are participation points where you have 5 participation points. There's 50 total, 10 per unit. You get 5 that I hand out in the class, 5 that you get outside of class, go into one of the review sessions. There's a Zoom recording of at least one of the review sessions. That would count for a review session for you to be able to take those participation points. Okay? Again, encouraging everybody to study before the exam. Okay, so why do I have this picture up here? Any ideas? Huh? I think they look like trees. And I think, you remember how I've talked about like the architectural formation or the microarchitecture of, say, nerves and bone and muscle being very, very similarly organized, right? These cylinders in bundles. So biology likes to conserve things. Biology figures out a way that it works. It kind of sticks with that system. It just kind of makes sense, right? You look around the room, it's winter time now. I think fall is pretty much over. Everybody would agree, right? It's winter, and there's winter clothing. You know, you've got vests and hoodies and puffy jackets and, right, thicker boots and mittens and gloves. And for the most part, yeah, there's different styles, and every 10 years or every 20 years, there's like a new look and feel to a mitten or a glove. But essentially, it's either a glove that has 5 spots for your fingers to go, or it's a mitten, right? I mean, that really hasn't changed. Oh yeah, if you live in certain areas, you know, the tops of the mitten roll over and your fingers are exposed so that you can actually do something with them. That's a new twist. But at the end of the day, it's still a mitten. So we even do that as humans. We actually conserve things that work. And so I think if you look here in Flagstaff, that's up at Snowball, you'll see this architecture that reminds me of neurons. And it shouldn't be a surprise. Like I was in Missouri. We were on a fishing trip, and there's no aspen, but there's a lot of juniper and oak, lots of sagebrush, a lot of trees that are deciduous trees losing their leaves. Same kind of thing. And in the wintertime when the trees are off, there's a main stalk, and there are branches that come out. And that's exactly how this neuron is organized. You can kind of appreciate the cell body in the middle, and you can see the dendrites that extend out, this axon. You can see how they synapse onto one another. I think they link up. And aspen trees, one of the reasons I picked these aspen trees actually have these underground root system that connect one aspen to another. And it's really quite amazing, but also annoying if it's in your front yard and you only want aspen in one area. Okay, but they're connected like neurons would be connected. Okay, so biology conserves a lot of these principles. And we do the same thing, right? I mean, for the most part, how many wheels does your car have? How many tires? 4. Anybody drive a truck that has 4 back tires, like a dually? Okay, that's a little variation. Or maybe one of those fancy motorcycles that has like 3 wheels, right? Car, motorcycle, I don't know what it is. You see them in the summertime up here. I don't want to see them right now. But for the most part, What's the shape of the wheel or the tire? Round. It's been round for a long time, right? It works really well. Or a steering wheel. What's the shape of a steering wheel for the most part? They're pretty much round, okay? So we do the same thing. So when you look at these things and you can see patterns that become familiar, I think it makes it less intimidating. Because I know that nerves and nerve physiology can be intimidating. So I'm going to be flashing back to a lot of stuff that you already know in this lecture to try to encourage you that this isn't as much new stuff than you think. A lot of it you've already been exposed to. A lot of it you actually already know. So see if you can, let's just practice what you know. Let's see if you can draw a typical cell. Somatic cell just means a body cell, like a generic cell. It can be a circle. That's fine. You could make it a neuron. You could make it a muscle cell. It doesn't matter. It's at rest, so it's at resting membrane potential. The membrane we're talking about is the phospholipid bilayer. And I want you to highlight 3 ions: potassium, sodium, and chloride. So potassium is K+. Back to unit 1, we looked at the periodic table. Sodium, Na+, and chloride ion, Cl-. Now, who can tell me by what mechanism are these ions under resting condition moved? What mechanism moves these ions under resting condition? Anybody answer that question? You know it. What's that? It's not diffusion, but it's a good guess because it's going to play a role here in a second. That's the answer to the next question. So if it's not diffusion and it takes ATP, it's an active transport mechanism we've learned about. What is it? It's a pump of some sort because it takes energy, just like any pump that you plug in the wall and has to run. Sodium, potassium, we call it the ATPase pump. It's ATPase, it's an enzyme. The enzyme is what breaks down the phosphates off of ATP. You learned all about that in muscle physiology, how 3 phosphate molecules on ATP, every time a phosphate comes off, it liberates energy. So the ATPase word is the enzyme that cleaves those phosphate molecules off of adenosine triphosphate. Right? You already knew all that, but now we're kind of just synthesizing some information. So you have a pump that's moving these ions. All right. In your drawing, can you tell me where sodium at rest is high, where potassium at rest is high, and chloride ion at rest is high? And this, by the way, is a great exercise to do on your test packet right when you get it. Like, just draw it out. So you can look at it and go like, okay, which way are the ions moving, right? Because in a second here, we're going to talk about diffusion. Well, if it's going to go down a concentration gradient, you kind of need to know where the concentration gradient starts. So I'm trying to help you learn the starting point. Where is sodium high? Outside the cell. Very well done. Where is sodium low? Inside the cell. Well done. Where is potassium high? Inside the cell. Where's potassium low? Outside the cell. Very good. Okay, this is the challenging question. Where is chloride ion high? It is high outside the cell. Very good. Okay, so what you're drawing is this. So you see this membrane, this transmembrane proteins, these 2 of them, an orange one and a blue one. But what they're stuck in is this phospholipid bilayer. So we just have a typical cell right here. It could be any cell in the body. It could be a nerve cell. Could be a muscle cell like we just got done studying. This is the intracellular fluid, the ICF, the extracellular fluid, the ECF. And you can see at rest, we're not showing the sodium-potassium pump, but you know that it's there. In fact, it wouldn't be a bad idea to draw it over here and just say, this is happening. And that sodium-potassium pump moves sodium out, right? How many does it move out? 3 out. and then it moves potassium which direction? In. And how many does it move in? 2. I would put that on here, right over here, to make this fully complete. If this is sort of your memorization cheat sheet, you can totally draw this on your test packet. There'll be some white space that you can draw this. And you can go back to it and look at it on some of the questions. Because you know Keller's questions, you're reading into the 5th paragraph and you're like, okay, I forgot what he was asking. I got to go back, right? You're like, so real. So, sodium channel, 5 paragraphs is an exaggeration, let's be honest. 2 is plausible, but 5 is excessive. Okay, so sodium, when this channel opens, these are channels. These are channels. In this particular diagram, this one is actually a voltage-gated channel. What does that mean to you? Electricity opens and closes it, just like a garage door, right? Or like a, that's a bad example, funny example, like a prison door, right? Where they, bzzz, you know, the prison doors in the movies. In the movies, you know what I'm talking about, right? I'm not saying anybody knows what those are firsthand, but you know, you've seen the movies. Like, we're all, you know, do you guys anybody watch the Lake of the Ozarks? Oh, here I go down a rabbit hole. Do you guys any watch the Lake of the Ozarks? So we were on Lake of the Ozarks, and so like everything was like, you know, about Lake of the Ozarks. My brother actually caught a blue catfish. He was like, blue cat, and he's like hopping around, and it was hilarious. But you know, like in that movie where like the attorney meets with their client, and they're like, we're finished in here, and they go bzzz, and the door opens. So an electric gate is what a voltage gated channel is. Back to the topic, right? This gate opens because electricity buzzes it open. Which direction does sodium go? Down its concentration gradient via diffusion with no energy required because the ATPase pump set up the gradient. Which way does it go? Which way does sodium go? Into the cell. Thank you. Perfect. This diagram is showing what we call a leak channel, a potassium leak channel, which is, it's always open. So back to the boat analogy. Anybody have any boating experience? Anybody know what a bilge pump is? What's a bilge pump? What does it do? Yeah, so all of the, if the boat takes on a little bit of water, because every boat isn't completely waterproof, it settles in the lowest point of the boat, in the hull, usually where the engine is. And so there's a pump that actually will pump water out of the bilge. And that's called the bilge pump. I'm going to close this. Just keep it nice and toasty in here. It's like a fireside chat. So because this is constantly open, there's a little bit of potassium that trickles out because the sodium potassium pump makes a high concentration of potassium inside the cell. So from the leak channel, it's kind of leaking out. This is one of the reasons that the ATPase pump is kind of always on, to keep the concentration gradient there. Okay? Someone's going to ask the question, well, that seems dumb. Why do you even have that then? Why don't you just make it like non-leaky? Wouldn't that be more efficient? That's a great question that I don't have the answer for. Okay? This is some of the stuff that it just is. There's probably some evolutionary significance that I don't know about. Okay? There's probably some mushroom in the African tundra that leverages replication by this potassium pump, and we inherit it and we don't need it anymore. I just made that up. Okay? So there's also voltage-gated potassium channels. Just like the voltage-gated sodium channels, that they hit electricity hits them, they will open. Okay? They're going to be more specific, obviously, and under more control than the leak channel. So you can kind of appreciate the relative concentrations. You see, I am not going to ask you on the exam, you know, I'm not going to put like 140 milliequivalent of sodium, 142, or is it 145? A, B, C, right? I'm not going to do that. I want you to know where is it high and where is it low. You don't need to memorize the numbers. For those of you that are really curious, I mean, these are the actual concentrations, but you can see sodium is high outside, potassium is low, chloride ion is high. Sodium ion is low, it's 12 versus 145, same units. Potassium inside is 150 versus 4, same units. And chloride ion is 5.5 versus 110, same units. Okay, so I just want you to know where it's high and where it's low. Okay, now look at the cell that you drew, or if your cell doesn't look anything like that, you can, the cell on this picture, you can just refer to that one. I want you to pair up, and I want you to talk about some examples of ions that use these mechanisms to move. We talked about voltage already. We talked about voltage gates. We talked about channels, like a leak channel. That's what that is. Now, I want you to give examples to each other. A, you have a ligand channel, and person B, you have a mechanical gate. See if you can come up with examples. Okay? Partner up. You can do it in threes if you need. That's fine. Do it in threes or fours. I heard you. Perfect. All right, person A. I need a person A volunteer of an example. You can give me another example of a voltage-gated channel if you'd like. If anybody have one? We studied this before. We studied it in a prior unit, a voltage-gated channel. Other than sodium that I just gave you, right? Think about muscle contraction. What's the molecule, the single molecule that regulates whether it contracts or not? Calcium. That's a voltage-gated channel stuck in the sarcoplasmic reticulum. And electricity, remember, goes down the T-tubule. You're like, oh, crap. Yeah, that's right. See, same motif. Like, you've already learned these things. And the body uses the same stuff over again. Like, why reinvent the wheel? You know that saying? That's why we say that. Okay, what's a ligand example that we've learned about? We've studied it already. I heard you, that's why.
When the ACH goes across the synaptic cleft and it binds to the sodium potassium gate, and then it releases.
Well, it binds to its acetylcholine receptor.
Oh, yeah. Right.
And then it opens a channel, right? It opens that channel and sodium's allowed to come in. Yep. So it's not electricity in that situation. It's a ligand. The ligand is acetylcholine. So sometimes this terminology throws students off. They're like, a ligand-gated channel? We've talked about that, Keller? We've talked about it. I described it differently. It had a different name. It was an acetylcholine receptor. But the concept was a ligand-gated channel that opened and sodium rushed in. Okay? So you've already seen that one. Okay, person B. Give me a... we talked about the leak channel. So you're off the hook on that one. Talk about a mechanical gate. And a volunteer in person B. So we've alluded to this. We haven't studied in great detail. Okay, so person B's had the harder challenge. Nobody came up with anything? So a lot of sensory receptors would be mechanical gates. Sensory receptors. We did talk about that. We're going to see it more specifically in the next unit when we look at special senses. okay? Because we're gonna see things that respond to like vibration in the inner ear. But you've seen like Meissner's corpuscles and Ruffinian corpuscles in the skin in lab, right? So these are all mechanical gates where there's pressure and the physical pressure triggers an action potential. So you literally can flick a nerve, right, and stimulate it by putting pressure on it, okay? We saw this when we looked at the occipital lobe and seeing the stars when athletes fall and hit their head, right? Jake Paul and Mike Tyson, neither one of them fell because neither one of them were fighting. But I thought maybe somebody might slip and then hit the back of their head, and that would be a stimulus in the occipital lobe, and that could trigger an action potential mechanically. Okay. All right. So it sounds like we're all on the same page on that fight, huh? I was worried I was being overly political, but it sounds like there's 100% agreement. Okay. I saw a meme. Now I'll probably get a little overly political, but I saw this meme of Jake Paul's next fight. It's before the actual one came out. It was like 5 minutes after the fight ended, and it said that he had challenged Jimmy Carter to a fight who's turning 100 this year. So then he could say that he beat a president. But anyway, okay, bad joke. All right, I won't do any more because there might be some fans, Jake Paul fans. All in good fun. All in good fun. He has the last laugh. I mean, he did make $40 million on that whatever you call it, that performance. He made $40 million. That's pretty good. Okay, back to this. Lake of the Ozarks, Mike Tyson, Jake Paul. Which is higher in concentration inside a typical somatic cell? Somatic is just body cell. Which one's higher? Potassium. We just got done talking about that. What causes a cell to be mostly negative? What causes a cell? This would be in the reading if you did the reading. But what's responsible for causing the cell to be negative? The inside of the cell. Not like negative in its emotional status, right? But the inside is electrically slightly negative at the surface. Nucleic acids are negatively charged, and proteins are negatively charged. There's a lot of DNA and RNA inside of a cell. There's a lot of proteins inside the cell. So the best answer would be 2 of these. So the inside of the cell has this slight negative environment. The sodium-potassium pump helps to create a gradient that's slightly negative at the surface, but just at the surface. And it does that by shuttling out 3 positive ions and only bringing in 2 positive ions. So there's a net loss of 1 positive ion. Did you catch that? So if I take 3 positives out and I only bring 2 positives in, I know we're not mathematicians, but that's 1 less positive, which is slightly negative inside the cell. You all follow that? I know physiologists, we don't like math like that, but that's how that works. So that's the sodium-potassium pump helps to maintain this slight electrically negative environment right at the surface with ion exchange. So subtle differences, but on the exam, don't be caught off guard about what the ATPase pump does. It doesn't create the negative environment inside the whole cell. It's just at the surface, and it helps to maintain it. It doesn't create it. It doesn't establish it. That's happening by other contributing factors. Okay, let's look at this animation. Ooh, fun.
unstimulated, neurons maintain a constant electrical difference or potential across their cell membrane. This potential, called resting potential, is always negative inside the cell. It ranges from -40 to -90 millivolts. If a neuron is stimulated, the negative potential inside the neuron can be made either more or less negative, depending on the stimulus. If potential is made sufficiently less negative, it reaches a level called threshold, and an action potential is triggered. During the action potential, the neuron suddenly becomes 20 to 50 millivolts positive inside. Action potentials last a few milliseconds before the cell restores its negative resting potential.
Okay, so we're going to talk a little bit more about this. I just wanted you to see this video so you can kind of put something to motion as we look at still imagery. Okay, and we're talking about a lot of nomenclature, right? So, for example, like this narrator said, you can make the inside of the cell less negative. Another way of saying that is make it more positive, right? Right? So sometimes the nomenclature tricks students unintentionally. So you just need to kind of pause for a second, maybe write it down and think through. If the inside of the cell is negative under resting conditions, and in order to send an electrical signal down the cell, you have to disrupt that slight negative environment. You disrupt it by making it more positive in that location. Okay? And that more positive spot with the movement of ions, we'll talk about this, it propagates downstream because the next area, the channels open and close and move those ions to make that next area of the membrane more positive. We'll talk about what happens behind so it doesn't go backwards. It only propagates forwards. But this is the concept. We just move ions. We move sodium and potassium ions along the surface of the membrane to create an electrical signal, which is kind of hard to think about, but you understand that they're electrically charged ions, right? They're +1. So if you move 3 in one direction and only 2 in the other, you create a negative environment inside the cell. And then if you move sodium in and you move potassium out at different times, you can change the electrical potential of that particular location. And electrical potential is just terminology that means electrical difference. Electrical difference. Okay, so the resting membrane potential of a cell is due mainly to what? Open sodium channels, open potassium channels, saltatory conduction, differences in calcium concentrations across the membrane. Did we talk about calcium differences? No, it's not that one. The only channel that's really open at rest is the potassium leak channel. So that would be B, correct. Okay, we're going to do this today because we only have this week. I want to make sure that I oversee it with you. I want you to create a chart down here if you don't have the slides with you, and you're going to put the word into or the word out of in the box. Into or out of. And I'm giving you the information that we saw on that deck, that slide. I'm giving you the actual intracellular fluid concentration. That's what the brackets around ICF stand for. And then I'm giving you the extracellular fluid concentration, respectively for potassium, sodium, and chloride. And I'm only focusing on sodium and potassium. So the diffusional force of sodium at rest is the diffusional force bringing sodium into or out of the cell. What goes in that first upper left box? How many of you say into? Raise your hand. Raise it high. Okay. How many say out of? Okay, good. How many of you don't know yet? You're going to wait to vote until after you see the answer? Okay, fair. A couple of honest folks. Into. Why is it into? Why is the diffusional force into? Sodium's high where? Outside the cell. It's low where? Inside the cell. It's going to go down a diffusion gradient or a concentration gradient, flow into the cell. Okay. Diffusional concentration force into or out of the cell for potassium? Out of, right? Out of. In the bottom left. The electrical force. Now we're talking about electricity. You're like, wait a second. Remember I said a potential is just an electrical charge. Is the electrical force of sodium into or out of the cell? At rest. What's the charge of sodium? Molecule. What is it? Positive. It's +1, correct? +1. What's the electrical charge of the inside of the cell at rest, especially at the surface, under resting conditions? Positive or negative? Negative. Opposites attract, right? So sodium is going to come into the cell down an electrical force. It's attracted to the inside of the cell because it's slightly negative, and it's a positively charged ion. What about potassium? Electrical force at rest. Is it into or is it out of? What's the charge of potassium? +1. What's the electrical charge of the cell? Negative. It's into. Some of you are like, well, that doesn't make sense. It leaves. It does leave, but in relation to sodium, do you think sodium comes in faster or more aggressively when it's able to? Because it's going down its concentration gradient and it's coming down an electrical gradient. And potassium is moving out of the cell down a concentration gradient, but it has kind of, it's like leaving, and it's like, wait a second, it's kind of nice back there. Okay, because it has an electrical pull back into the cell, but it's high concentration in the cell. The diffusional force overpowers it. So potassium, you're going to see, is going to move more slowly. Sodium's going to move very fast, and this is one of the reasons, and that's why I'm setting it up this way. Okay? Does that make sense? So rather than have you marinate on that overnight, and come back on Wednesday, I just thought we would go through it and I would explain it today. Okay, so here we go. You've seen this before, right? So what's an action potential? Who can tell me what an action potential is? You guys have watched the lecture, you've done the reading, right? So who can tell me what an action potential is? It's a depolarization event. What the heck does that mean? What is a depolarization event? See, now in this class, we're starting to define things with other words that we have to define. That's why it's really important for us to stay on top of the material and the vocabulary. What does that mean? That's totally correct. A reduction in membrane potential. How about just an altered membrane potential? Okay, altered membrane potential. The membrane potential becomes inside the cell more positive or less negative. It crosses a critical spot known as threshold, minus 55 millivolts, and the neuron fires a signal. The neuron transmits electricity downstream. That's the definition of an action potential. That's the definition of a depolarization event. To depolarize means if you have something that's polarized, so the outside of the cell is positive, the inside of the cell is negative, it's polarized. They have different viewpoints, right? In the last election, in all elections, most of the candidates are polarized. They have different viewpoints on topics. Depolarization means that you make the outside and the inside less different. So you make the inside less negative, making it more like the outside. You're depolarizing the membrane. That's why it's called that. A lot of terminology here. What's a local potential? Another word for it is a graded potential. How is that different than an action potential? What's a local or a graded potential?
It's when there's still a stimulus, but it doesn't quite hit that -55, so it doesn't cause a whole action.
That is correct. It's a stimulus. There are channels opening, ions are moving, but not enough flow is happening where the membrane potential doesn't hit -55 millivolts. It doesn't cross threshold, so it never becomes an action potential. It stays as a local graded potential. Okay? Why the difference? Well, so one of your colleagues was in class in office hours today, and you guys have played the hot hands game, right? So someone could come up here and put their hands on top of mine, right? And your reflexes will be way faster than this old guy, right? But every now and then I might get you, right? I might, you know this game? So when you're getting ready and you're sending signals, you know, and you're about ready to fire, that's where you're getting these local graded potentials. But you can't like turn and go and really stop once the action potential fires. Once you send the signal to contract the muscle, it's over. It's an all or nothing on an action potential. But a graded potential, it just primes the system really close to threshold so that you're faster at slapping your friend's hand. Okay? Kind of the best example that I could use. So the action potentials only occur where there's a high concentration of voltage regulated gates. Voltage regulated gates. They have a high concentration in the trigger zone, which is the axon hillock, the neck region of the neuron. That's where they centralize. The cell upregulates the expression of these voltage-gated, voltage-regulated gates at that location. And so an action potential can be caused by a local potential. So here we have what we call excitatory postsynaptic potentials. So obviously we're past the synapse. That's why it's called a postsynaptic potential. It's excitatory because you've got a stimulus and it's depolarizing the membrane, and you can stack them on top of each other until you reach threshold. Once it hits -55, it triggers an action potential and it's gone. I'll tell you, there are also what we call inhibitory postsynaptic potentials that actually make the membrane more negative, further away from threshold. They're inhibitory postsynaptic potentials because they make it less likely that an action potential is going to fire. So there are pharmaceutical agents that will do this, that will desensitize the nerves. And see, these are some of the mechanisms of how the pharmacology works. You have hyperactive nerves, you want to desensitize them. You have more inhibitory postsynaptic potential, so the nerve's not sending a signal as easily. So this diagram, this -55, then we come all the way up to +35. And then we repolarize the membrane, bringing it past threshold, past resting membrane potential. We actually overshoot, and then we reestablish resting membrane potential with the sodium-potassium pump that actually resets the ions exactly the way they're supposed to be. And in this period of time, on this overshoot, or what we refer to as hyperpolarization of the membrane, that's where that membrane won't fire again. And by not firing in the spot you just left, it allows the propagation of electricity to go forward instead of going retrograde. That's the reason for the refractory period. Okay? So let's talk about some of the specific events that are happening at these different stages. Right? You should absolutely expect a test question on these events. So number one, we're in the phase here of building up to minus 55. So we have local potentials that are building. I actually think this is a better representation of really how it works. But for simplicity, we're just showing this increasing line. It could be one signal, but usually it's multiple signals. Okay? But for simplicity, this is easier to study and understand. So in segment number one, sodium ions arrive at that axon hillock and depolarize the membrane at that point, because sodium is rushing into the cell fast. The threshold is reached at minus 55. Now, voltage gated or voltage regulated channels open at minus 55. We call them fast gates because they open quickly and sodium floods into the cell. As it floods into the cell, bringing all that positive charge into the cell, more negative is at the bottom, more positive is at the top. This is why this spike is so quick and so fast. This is time. Right? So at step 3, this is where the fast gates open. You also have in the background voltage-gated potassium channels, which are called slow channels. They're slow to open, and potassium is slow to move because it's fighting against an electrochemical gradient, right? The electrical force is back into the cell, and it's leaving because it's going down a concentration gradient out of the cell. So in the background of 3, the slow voltage voltage potassium-gated channels open, but the effect is drowned out by the sodium gates that are bringing in so much sodium. At 4, this is when the sodium gates close at 4, and they close shut. As soon as they close shut, the potassium gates that opened at 3, but they were slow, stay open and are responsible for, bless you, the repolarization event, where you're getting sodium no longer moving, potassium is leaving. If potassium is leaving down its concentration gradient, it's taking positive charges out of the cell, making the inside of the cell more negative. So it outflows potassium. They're fully open at 5. The potassium slow gates are fully open. They're also slow to close, and instead of having an abrupt close and then you just hit resting membrane potential, like here, this was an abrupt close with the sodium gates. The slow close with the potassium gates allows for this hyperpolarization. You overshoot. You allow too much potassium to leave. And then you just clean it up with the sodium-potassium ATPase at 7, reestablishing resting membrane potential. Does that make sense? So I would like you to be able to explain the 7 different stages and steps of what's happening along an action potential. And some of the key points that are labeled and the key things that we just walked through are totally fair game on the test next Monday. Questions? Up front.
Are there any clinical examples of where that hyperpolarization step doesn't happen and causes problems, or does that not really?
No, the question was, are there any clinical examples of, missing the hyperpolarization? I don't know of any. I mean, I'm sure we could Google Scholar it. Your bonus opportunity is going to be reading about multiple sclerosis. It has nothing to do with this, but it does actually have to do with how the signal is propagated. And in MS, there can be some problems with efficiency on the depolarization being propagated forward. It's typically attributed to the loss of myelin and the inefficiency of the signal kind of hopping from node of Ron VA to node of Ron VA. And I'll explain that here in a second. So there could be some retrograde signaling that's lost. And MS is a progressively debilitating, so it gets worse as the patient gets older. So I wouldn't be surprised in certain cases of MS if you see that, but that would be the only one that I would really know off the top of my head. Great question. Okay, so characteristics of an action potential. It's all or nothing. Once it hits threshold, it fires. If it doesn't make it to threshold, it doesn't fire. It's non-decremental. That means it doesn't get smaller. It doesn't dissipate over time. It doesn't wear out. Once you send the signal to fire with fidelity, like with a pure signal, it fires all the way down the nerve. It doesn't dissipate. It doesn't wear out. MS is a different story. Okay? It's irreversible. Like, you can't change your mind. Think of the hot hands example, right? You're really trying to, slap, and you're going to miss, and you can't stop it. It's too late, right? Even your antagonistic muscles can't pull it back fast enough. So it's not reversible, the action potential, whereas a graded potential is. It is definitely reversible. And a graded potential is not all or nothing. It actually dissipates. If you don't use it, it wears out. If you don't reach threshold, the local potentials, the graded potentials fade, and they're not used. So after an action potential in this refractory period, we kind of have two different phases of this refractory period. We have what's referred to as an absolute refractory period, and then we have a relative refractory period. So the absolute happens when you see here the neuron is actually firing. When an action potential is actually happening, you're not going to fire another one. And it lasts essentially until it hits resting membrane potential. And then you're in this relative refractory period where, I told you it prevents it from going backwards, the signal. It only goes forward. But if you get a signal that's of significant strength, you can actually still fire an action potential in the relative refractory period. The absolute is absolute. The relative refractory period, it takes a signal of significant strength more than what you normally would have for it to fire. Okay, I do want you to do this one at home. Compare and contrast, and I'll tell you the answers, or I'll post the answers in the announcements. Action potential on the first column, a graded or a local potential on the far right column. All or nothing, is it decremental? Can it be inhibitory? Is it reversible? Short or long-lived? Vary in strength? And where does it happen? So anticipate some questions on the exam about the differences between action potential or a local, otherwise known as a graded potential. So next up, let's talk a little bit about propagation of the signal. So these diagrams of these cylinders, let me explain them for a second so that you can kind of better understand what we're talking about. But if you look at this box, can you guys see this cursor? Okay. If you look at this box right here, that's the area that we're focusing on in these three little cylinders. And this is representative of this part of the axon. If it's in green, it's resting. It can be excited. It can stimulate an action potential. If it's in yellow, it's refractory. It's already fired, but it's relative refractory. If it's red, an action potential is actually happening, and it's an absolute refractory period at the moment, right? So hopefully what you can see on the movement of ions and changing at the surface of the cell, the negative environment to being slightly positive, which is indicated by this action potential in red happening. Right here at the interface, it's positive inside the cell. And as this moves to the right, this positive moves to the right, you literally are sending that electrical signal down to your right. just by the location of those ions at the membrane surface moving. When you open a voltage-gated channel and sodium moves and then potassium moves. So if the nerve is unmyelinated, it's called continuous conduction. It moves all the way down the nerve. Every little millimeter of the nerve has voltage-gated channels that are opening and closing. If it's unmyelinated, continuous conduction. If it's myelinated, the signal actually moves from the bare spots of the neuron to the next bare spot of the neuron. And where the myelin covering is, that insulation, there's no ion movement. So the signal just hops. It like literally leapfrogs across from one bare spot to one bare spot. One, what we call node of Ranvier to node of Ranvier. So it's much faster because you don't have all of those channels having to open. You only have channels having to open every time there's a bare spot. Make sense? So that's why myelinated neurons travel the signal much faster than unmyelinated neurons. So the two things majorly that impact the speed of conduction of a nerve is myelination, and we talked about that. We talked about that in prior units because we've looked at white matter versus gray matter. And a lot of the white matter tracks were tracks that were actually moving from the left to the right hemispheres, right? Large tracts of information, sending lots of information, or moving from the central nervous system, from the brain all the way down the spinal cord so it can get out to the periphery. And at the local level of those inner neurons, there was no myelination. We didn't waste the biological energy making myelin. But we have long tracts, or we need things moving really fast. We go ahead and manufacture the myelin that's made by which cell types? From the last lecture, which cell types make the myelin? Which glial cells make the myelin? In the central nervous system, it's the oligodendrocyte. And in the peripheral nervous system, it's what? Schwann cell. And you're going to learn about those in the paper that I'm going to give you for the bonus opportunity. You're going to learn about this in more detail. Well, the second biggest parameter of influence of the speed of conduction is the diameter of the nerve. So it's like, think about drinking a milkshake. Small diameter, can't get a lot of milkshake. Large diameter straw, better, right? Brain freeze, way better. Okay? So the larger the diameter diminishes the interference or the resistance of the signal. So here's a graph that shows fiber diameter across the x-axis and conduction velocity across the y-axis. And so you can appreciate that unmyelinated versus myelinated, they're completely different ball games, right? The myelinated fibers have a much faster conduction velocity, but the fastest would be a large diameter myelinated fiber is going to give you, 100 meters per second. And if you have an unmyelinated fiber that's large diameter, say all else being equal, you take a 20 micron diameter fiber. If it's myelinated, it looks like it's at about 120 meters per second of conduction velocity. If it's unmyelinated, it's only at about 10, right? So large diameter and myelinated, fastest speed. That's the take-home message. All right, let's watch this video.
Conduction through myelinated nerve fiber. During myelination of neurons, action potentials may only occur at the nodes of Ranvier. Myelin is made of insulating cells, which means depolarization cannot occur in myelinated regions. Between these cells, however, there are gaps known as nodes of Ranvier, which are unmyelinated. As depolarization cannot occur at the cells making up the myelin sheath, The wave of depolarization can only occur at the nodes of Ronvier. Thus, action potentials appear to jump from node to node when traveling down an axon, and this phenomenon is known as sulfate reconduction. And it serves as a means of increasing the rate of propagation of an action potential. Not only does sulfate reconduction increase the speed of impulse transmission by causing the depolarization process to jump from one node to the next, it also conserves energy for the axon as depolarization only occurs at the nodes and not along the whole length of the nerve fiber as in unmyelinated fibers. This causes up to 100 times less movement of ions that would otherwise be necessary, therefore conserving the energy required to re-establish the sodium and potassium concentration differences across the membrane following a series of action potentials being propagated along the fiber.
All right, great. We're not gonna watch all the rest of the show, but you kind of get the idea.
What you will see and hear in this presentation is the development of myelin in the peripheral nervous system and the propagation of the action potential along a myelinated axon.
I'm not sure why the audio is always so weird, but they are.
Good understanding of the Schwann cell and the electrochemical process of the neuron called the action potential. The Schwann cell forms a protective covering around the axon.
Do you see how it wraps its plasma membrane around?
The axon through childhood. This development increases the thickness of the wrappings, which peaks in adolescence. This is why teenagers have such quick responses.
This is the cell nucleus right here. See?
The Schwann cell contains the typical cell organelles and cell membrane structure. However, notice as the Schwann cell surrounds the axon that the nucleus and other organelles are squeezed to the outside wrapping of the cell. This outer wrapping of the Schwann cell is called the neurolemma. The inner lining is made up of layers upon layers of cell membrane. This inner wrapping is called the myelin sheath. You will recall that the cell membrane, called the fluid mosaic model, is made up of a bilayer of lipids integrated with proteins. The thicker the myelin, in other words, the more layers of cell membrane making up the myelin, the more advantageous it is to the axon. One advantage is the regeneration of severed axons. Another advantage is an increase in the speed of the propagation of the action potential along the axon. The rest of this presentation will concentrate on the increased speed of action potentials down the length of the...
Okay, we already covered that. Let me clarify on the regeneration. That only happens in the periphery. Okay, we have not been able to figure out why, but it's not well conserved in the central nervous system. This is why peripheral nerves regenerate fairly well. Central nerves like spinal cord injury patients do not. Stroke patients do not. So the concept here, just another view of it, same information, just a different slide. So this explosion-looking spot is actually sodium ion flowing into the cell, and this is only happening at the nodes. It's actually a French name, so it's not Ranvier, it's Ranvier, okay? So it's the nodes of Ranvier from one node to one node. In fact, you only manufacture voltage-gated sodium channels in this cell and insert them at the nodes. You don't put them underneath the myelin because there's no need for it. And you see this saltatory conduction, the action potential in red again is kind of hopping from one node to the next as you move down the action potential signal. So multiple sclerosis, what happens? You're going to read a paper on this in more detail. I think it's going to be quite interesting. But multiple sclerosis is actually an autoimmune disease. It's an autoimmune disease. It's a hypersensitivity where your body attacks your myelin and destroys it. And it happens over time. You destroy the myelin thinking that it's foreign and get rid of it. And so you change from a saltatory fast conduction to a slower continuous conduction. And if these are in motor neurons, then the patient's motor capabilities diminish as they get older. If it's in sensory neurons, their sensory information diminishes as they get older. And so in this paper, what you're looking at is the NAV 1.6 in red is the sodium-potassium pump, and an associated protein on either side is shown in green. So as you get the different stages of MS, you can kind of appreciate in this diagram, these are the voltage-gated sodium channels. This is the myelin. So in the start of a healthy neuron, you only have the voltage-gated channels in the nodes. You see the red with the green associated protein, the Casper on either side. As you lose the myelin, the body's compensatory mechanism is to insert new voltage-gated sodium channels so that you can still send an action potential, but it's just slower. So you start upregulating, you start inserting, you see this? You've inserted it here. Now look at this picture of this immunohistochemistry. Much more red in this MS neuron that's been demyelinated. And this continues. And eventually it continues where the nerve just can't keep up and you start losing the battle with the immune system that's attacking your myelin. Make sense? So I think you'll find this paper that kind of talks about how this immune system is tackling your myelin sheath a little bit interesting. So stick with me today. So axon diameter, as we kind of wrap up today's lecture, we'll get into some neurotransmitters at the very end and a couple of sample test questions. But the axon diameter, the larger the diameter, the what? The faster the speed of transmission. You have a less resistance to current, a less resistance to flow with a larger diameter. Think of it like a milkshake straw. If any of you have done any like car audio or stereo work, or have you done any electrical, if anybody have like a hot tub, you looked at a wiring a hot tub, look at the plug of the hot tub or the dryer. The wiring is huge, right? A hot tub plug, if it's a plug-in, it probably shouldn't be. It should be hardwired. But the dryer plug is a 220, and the hot tubs run on mostly 220 voltage. So the larger the diameter of the wire, the larger the diameter of the speaker wire, the better the sound, the more electricity you can move. It's the same thing with neurons because they're basically like our wiring, our biological wiring. So the larger the diameter of the wire, the larger the diameter of the neuron, the faster the speed of conduction. Okay, does this figure look familiar? Okay, so we're just going to finish this thing out, kind of going back to the synapse. You know, and in prior semesters, we would do this first and then muscle, but you've already learned about the synapse. So, you know, I don't have to talk about all this terminology. You already know it. We've got our presynaptic side. We've got our postsynaptic side. We've got an acetylcholine molecule that moves across as a neurotransmitter. Acetylcholine is up here, ACH, acetylcholine, neurotransmitter. You know it really well. It crosses the synaptic cleft, communicates with the postsynaptic side. Well, there are other neurotransmitters. There's over 100 known different neurotransmitters in our body. We've got acetylcholine. We have GABA, glycine, aspartic acid, glutamic acid, histamine, serotonin, dopamine, epinephrine, and norepinephrine. You guys have seen those actually already. We've got neuropeptides, enkephalins, substance P, which is responsible for pain. That's the neurotransmitter that goes through pain pathways that give us information about pain. Cholecystokinin, endorphins, all of these structures are neurotransmitters that allow us to either be excitatory or inhibitory. Now, I'm not as interested in you memorizing all of these. I think it would be important for you to be very familiar with acetylcholine. We've studied it in great detail. I would be very familiar with epinephrine and norepinephrine. Then what I would like you to do is be able to classify how they work. So they're either referred to as being ionotropic or metabotropic. If they're ionotropic, it's because they alter the membrane potential. They influence the ions. So give me an example of an ionotropic neurotransmitter that you know really well. The one we've studied the most. Acetylcholine ACH. It's exactly right. It is an ionotropic neurotransmitter. The rest that don't alter the membrane potential are what we call metabotropic, because they use some second messenger system inside the cell to impact some metabolic process. So they're referred to as metabotropic versus ionotropic. That's a hard word to say sometimes. Okay? All right. Saltatory conduction. Action potentials hop or jump from where to where? From one Ranvier to another. Questions? Comments?
Great job.
All right, great. That's awesome. All right. I'll have office hours on Wednesday. Appreciate the clapping. That's awesome. I mean, that doesn't happen every day. I did have a vacation last week, so. But we're not over. We got one more lecture on Wednesday, right? So we'll finish up. Oh, Keller. You take back the clap, right? Okay, pause for a second. You're going to need this on the screen here.